Hello, this is Sudharshan, and in this episode, chapter 42, we will talk about cholera, Halophilic Vibrio, and Aeromonas infections. So, let us start with cholera. So, cholera is caused by the microbe that is Vibrio cholerae, which is a gram negative bacilli. That means it is a rod shape and doesn't stain gram stain, and it is comma shaped mainly, and apart from that it is aerobe, non-motile, non-spore forming, and fastidious. Yes, and with respect to the now we will see the mode of transmission. It is fecal-oral route, and with the risk factors, mainly persons with O blood group, and commonly malnutrition, low immunity, poor hand hygiene, sanitation measures, and all. Alright, and with respect to the pathogenesis, so we will see now. Right, so the pathogenesis is mainly through the cholera toxin. That is how it is going to act. So this toxin will have two parts, A and B. So the A1 causes ADP ribosylation that activates the GS subunit. This is very important. ADP ribosylation, where ribose is added to ADP, and that increases the GS. So S is stimulation. So cyclic AMP increases, and the B binds to the GM1 receptor. That is very important. And because of that, there is chloride retention and reabsorption of water and sodium. And apart from chloride, even other electrolytes are also retained. And with respect to the clinical features, the most important for cholera is rice water stools. Right, and apart from that, there can be GI distress, then musculoskeletal abnormalities because of electrolyte imbalance. Their features will be there. Right, and with respect to the diagnosis, let us see now. Diagnosis, sample collection, mainly we will do a stool sample collection. And with the culture, we can see the darting motility of the bacteria and also comma shaped. And also we can go for selective media like TCBS, that is thioglycolate, bile salt, sucrose agar. That is the TCBS agar, where we see the yellow colored colonies, and on blood agar, beta hemolytic colonies will be there. Apart from that, McConkey agar as well. And then serological tests like ELISA PCR, antigen antibody testing, Malditoff Whitek. Then biochemical tests, it will be urease negative, citrate catalase oxidase positive. It doesn't form hydrogen sulfide. It is lactose fermenting but doesn't ferment sucrose, but it will ferment glucose. And then antimicrobial susceptibility testing. And what differentiates between Vibrio cholera and Campylobacter jejuni is that Vibrio cholera survives at a higher pH. And when it comes to the treatment, it is going to be supportive care, like managing the water and electrolytes, for example. Antibiotics would be reserved for severe infection. And with the prevention, obviously, like proper sanitation measures, good hand hygiene, eating well cooked food, and getting vaccinated for cholera. And cholera can also show extraintestinal manifestations. Apart from the GI, other systems also, other viscera will be affected. And then next we have halophilic vibrio infections. So from the name, halo is salt, philic is loving, vibrio is comma shaped. These are the microbes which look very similar to Vibrio cholerae. They have a comma shape, but they are salt loving. So we have the organisms like Vibrio vulnificus, Parahemolyticus, and Alginolyticus. And they also cause the intestinal and extraintestinal manifestations. Diagnosis being the culture, TCBS, where mostly we see green colonies, yellow colonies in case of cholera. And apart from that, we can have Malditoff Whiteegg, urease test will be there, positive. And the same for H. pylori, that will also show urease test positive. And the swarming motility on blood agar. And with the treatment, it is going to be supportive care for all the halophilic vibrio I am telling. And then lastly, we have Aeromonas infections. So here we can see it produces a toxin, enterotoxin, exotoxin, endotoxin, that causes the pathogenicity. And very important, Aeromonas is a species called Aeromonas hydrophila. It is a water-loving one, and it causes red leg disease. Red leg disease. Same clinical features, intestinal and extraintestinal manifestations, diagnosis done by culture, Malditoff Whiteegg, and the treatment is supportive care. So that is all with the cholera, halophilic vibrio, and Aeromonas infections. So thank you so much. We'll see in the next lesson.